Okay. So hi everyone, I'm Damiano Piovesan, professor at the University of Padova, and I'm going to talk about this software CAFA-evaluator. And the presentation is split into two parts. The first part is more generic, and the second is dedicated to CAFA participants. It's a little bit more technical. So the software is actually the evaluation, piece of code that calculates the evaluation in Kaggle. So actually, you submit the prediction, and it generates the three numbers, the weighted F-score for each of the aspects of the gene ontology, and then these numbers are combined into a single score to make up the dashboard. The software is available in a GitHub repository and is linked somewhere in the documentation of the Kaggle page. Well, actually, there are other tools to evaluate the predictions of function prediction methods. There is one developed by the Ontological lab, written in MATLAB, and there is another one implemented by either Freedom Lab, implemented in Python 3. The first have been used in CAFA 2 for sure, in CAFA 3, and I think also in CAFA 4. I'm not sure about that. And the other one has also been used in CAFA 3. I'm not sure about which is the difference between the two and which calculation has been performed by one or the other tool. One thing for sure is that the Python version is limited to the calculation of the F measure. So one question is, why yet another tool? So my answer to that question is that it dates back to an experience I had in 2019. So I was a former developer of a function prediction method which is called Inga Tosato, here in green, which was in the top 10. So we wanted to publish the article describing the method in NAD the same year. And of course, they asked us to provide some evaluation. So we were able to reproduce exactly the same curve, which was provided by the CAFA organizer. So in the end, we decided to change the ground truth and provided just an internal evaluation of the components in comparison with the ensemble prediction. So one motivation is to improve the reproducibility of the result, which of course doesn't depend only on the software, but also on the all the details about how things are calculated in CAFA. Another problem was the use of the software against the different ontology. So in CAFA 4, I've been invited to organize a sub-challenge for the prediction of function of intrinsically disordered proteins, or the prediction of intrinsically disordered function-specific proteins. And to do that, the solution at the time was to try to modify the Python version of the evaluation software. It was very difficult because internally there were a number of variables which were hard-coding references to the gene ontology. So gene ontology IDs and a number of things which are fixed in the code. So still, I'm not sure that the result I share with the participant are correct. So now I recalculated everything using the new software. I also updated the ground truth, so I will share the result very, very soon. The new result very, very soon. So to sum up, the limitation of the existing tools is that they are limited to gene ontology, and for the Python version to the human phenotype ontology. There are no options to control the calculation, and there is complex procedure to generate the results. So if you try to use that, you know that you need to put files in a complex directory structure. There are a lot of manual steps, and a number of assumptions are made at each step in the calculation. So, for example, at a certain point, you propagate the scores, and you call another piece, another another comment, and actually there's no way you can check that the scores are propagated correctly unless you check the file with another piece of software. And of course, limited documentation, but this is true for all software. So the major theme was about reproducibility, and so there are a number of questions which are not answered inside the papers. And namely, this is the list of things which I identified. So, for example, how do terms included in the calculation are cross-ontology relationships considered? So sometimes you have part of relationships which goes from one aspect to another aspect. And what about synonym? So if you submit a prediction which use synonyms for the Go term, so every Go terms can have one or more synonyms, or can have a synonym. What about obsolete terms? And also how scores are propagated up to the root of the ontology, how the information accretion is calculated. Now in CAFA, in Kaggle, you have an information accretion file, so it's very, very good, but we don't have that file for the previous CAFA, so it's also difficult to test if, for instance, semantic similarity is calculated correctly. And last, how is possible that the precision of the code curves are monotonic? I will show later. So major features of the software is that it's written in Python 3. Just use 3 dependencies, NumPy and pandas for the calculation, and matplotlib only if you want to generate the plots. Takes very simple input format. It's generic, so it can work with any ontology. It's not limited to the gene ontology. And the logic is very simple. So all the logic related to data, the dataset generation, baseline generation, generation of information accretion values, they're all delegated to external tools. So the software is just focusing on the evaluation of the prediction, so the calculation of the metrics. It's fast, it exploits topological sorting and parallel calculation, and it uses a relatively low amount of memory. So the memory use is just depending on the size of the ground truth and the number of nodes in the ontology. It calculates also weighted metrics, both for the semantic measures and the F measures. And there is a separated Jupyter notebook for generating the plots, so that you don't need to recalculate every time the evaluation, but if you want to modify the plots, you can just use the data you have already calculated. Okay, so this was the first part. Now I will go a little bit into more details. What is the input and the internal representation? So the ontology is represented as an agency matrix. So you have those represented the source node, which are the nodes at the bottom of the ontology, the leaves, let's say, and the columns are the targets. So the relationship between every node and its their parents. The ground truth is represented as a Boole matrix, which is M times N, where M is the number of proteins and N is the number of nodes. And then the third input is path to the prediction folder. So in the prediction folder, the software expects to have prediction files, and it processes these files one by one. And for each file, it generates another matrix with the same size of the ground truth matrix, but containing the score assigned to every node. Optionally, it also accepts the information accretion file to weight the predictions, and this is stored just as a vector of the size of the node of the ontology. And you see here the input format, the format of the information accretion file. So these are very, very simple file to generate. So why it's fast? So one thing we exploited, which was actually, I think, already implemented in the MATLAB version, is to use topological sorting. So the first step of the software, when it parses the ontology, is to sort nodes. based on their depth in the ontology. So you see here an example. So the first nodes in the resulting vector, sorting vector, are 4, 7, 5, 6, 2, 3, 1. This is very useful because when you want to propagate the score, you can just propagate following this sorting array, and the calculation can be performed in parallel for all the proteins in the prediction matrix. As I said, the aim of the method was to improve the reproducibility and to facilitate the use by the community. So the software is flexible in terms of parameters it can take to modify the precision recall curves, and plus it's flexible in terms of usability. For instance, if you want to run it on a cluster and you don't have access to the file system where the software is executed, you can write the result in a different folder with this option. And in the next slide, I will talk about these other options, which are the most relevant for the differences you will see in different precision recall plots. If you're not aware about how what choices are made in CAFA. So the parameters regards the inclusion or exclusion of root terms from the calculation, the way you normalize the macro average values of precision and recall, and the way you propagate scores in the prediction matrix, and the number of threshold you consider in the calculation. So essentially the number of points you will consider to generate the precision recall curves. So for what regards the normalization in CAFA, historically, these are the two formulas for the precision and the recall. For the precision, the value is normalized by the number of proteins predicted at given threshold. So it might change depending on the threshold and depending on the method. Instead, for the recall, the total recall for a given threshold is always normalized by the size of the ground truths for the total number of proteins in the data set. So I will show an example how the precision recall curves change based on the different strategies. Here you have an example. You have two proteins, and this is the ground truth for the two proteins. And here you have the prediction. So only one protein is predicted, and these are the scores, and the other one is not predicted at all. So in the first case, when you consider only predicted proteins, both for the precision and the recall, you have the best case. And you have the worst case when you only consider the entire data set. So here, this line, you see my mouse? Okay, this one here. So in CAFA, historically, you have something in the middle. You see the precision is the same as in the prediction strategy, and the recall is the same as in the ground truth strategy. And another effect is when you exclude the root terms, and generally you get a decrease in the performance. Here, the decrease is quite large. It's 10% because the size of the ontology and the number of proteins is the size of the ontology is very low. But I mean, it still will be relevant also for the standard CAFA evaluation. So another thing I wanted to show is the different strategies you can use for score propagation. So here you have an example. It can be weird because you have one prediction for this term at the bottom, and then prediction also for some nodes which are in the ancestry of this term. And in particular, you see you have this term here, which is predicted with a score which is lower than its child here. And you can propagate terms two ways. You can always take the max across the children, and this would be the result. Or you can propagate with this strategy, which is called, I call field. I don't know if it makes sense, but for instance, in this case, this term is not overwritten when there is no assignment going towards the roots. The score is propagated, and when there are two children with different score, I mean, the strategy goes back to max. So what is the effect in the precision record curves? So here you have an example. So the ground truth again and a prediction. Here the field doesn't change much, the actual prediction, because you have only this term. But you have here one term with 0.3, which is actually a negative term in the ground truth. And you see that in the max propagation, you just get two scores. This is similar to the previous prediction. So the difference between what I explained before is that you have these terms with the lower scores. So in the score propagation and the precision of the record curves, in this specific case, you will get a better F score for the field propagation and this type of prediction compared to the max strategy. And this is because at score 0.4, this term here, which is wrong, is excluded, whereas for when you get the F max, in this case at 0.7, this term is included. So this is a really specific case in which the predicting the ancestry with a lower score can be beneficial to the F max you get at the end. So another thing is, of course, yeah, I'm almost finished. This is to show what is the effect using a different resolution of the different number of thresholds, and you see also the area under the precision of the record curves can slightly change because you have more points. And the last slide, how you can transform precision of the record curve in its normal way to the way it's shown in CAFA papers. Essentially, you accumulate precision going back to the recall so that you never get precision at a lower recall, which is lower than the precision at a higher recall. So essentially, if you make a prediction where you have the maximum precision at high recall, you will get just a straight line till the lowest recall here. So I think this was my last slide. I would like to thank Davide Zago, one of my former students that implemented the first version of the software and did spend half of his thesis reverse engineering the CAFA precision record curves, and Ido and Pedra that actually dug into their memories and found all the details which are fundamental to reproduce the details of the precision record curves. And with that, thanks for your attention.
Thanks, very interesting talk. So the score propagation is something that you run before you calculate the final scoring?
Yes. I mean, there are people that submit predictions which are already propagated. In that case, I don't do nothing. I mean, I try to propagate, but automatically you find, I mean, what they do. And yeah, we every time calculate the propagation, both for the ground truth and the predictions.
And so which one do you use to run the CAFA?
In Kaggle, yes. In Kaggle, I mean, the list of parameters we use in Kaggle is providing documentation. And for what regards to propagation, we use the field, this one. I mean, there was, in previous CAFA, I mean, according to what Pedra said, there were clear requests to use this strategy. I mean, there are methods that want to predict parents' terms with a lower score. I don't know exactly why, but
So if one was participating in output, because it's more beneficial not to actually propagate the scores, right? Because you might do with a way similar to the max, and that would be detrimental for the score.
If you propagate with max, it doesn't matter because I will propagate with max. I mean, the fill behaves with max if you just provide the deep terms. If you provide something like this, so if you give me two terms and they are on the same branch, then the way you propagate has an effect.
Thank you.